Hi, and welcome back to neurobiology in 5 minutes or less. Right now, we're going to be talking about the differences and similarities between the umami receptors and the sweet receptors. We're actually going to start out up here at the top with the sweet receptors. The reason why we're comparing these two is because they're both class C receptors, and they have very similar GCPR. And so GCPRs are G protein coupled receptors, which basically means that as the receptor is stimulated, a G protein cascade is going to be activated. And so in our sweet receptors, we have this one, the T1R2 and our T1R3. And our T1R3 is actually the same with both the sweet and the umami, but the T1R2 is unique to our sweet receptors. That one does not exist in the umami. In the umami, we have the T1R1. So after our receptor is stimulated, and the receptor is stimulated by having a ligand bind, maybe the ligand binds here, or it binds up here, here, or over here. Wherever the ligand binds, and then activates this. The G protein is set off by having an GDP, which is originally attached to the alpha subunit of our G protein, and right here, this one is our alpha, and this is our beta gamma complex. So the GTP and the GDP, that would all be over attached to our alpha because it's by himself, so he needs a bit more energy, whereas the beta gamma complex, they have each other. And so then these two can go out and do different things. And one thing that they decide to stimulate and work on is over here with PIP. So PIP is going to be located along the membrane, and PIP is going to be transformed via these two catalysts along with a whole chain of other reactions into IP3, and we have our proton. The proton is just going to kind of float off and do its own thing, but over here, the important thing is that we have our IP3. The reason why IP3 is important is because we're going to then use that IP3 to activate an IP3 receptor. And so the IP3 receptor, which is going to be located right over here, this is actually going to allow the influx of calcium, right? And so the calcium is going to be coming into the cell, and as the calcium comes in, calcium is going to come over here and stimulate yet another receptor, and this receptor is going to be a TRPM5. And this receptor is going to allow the influx of sodium. And so, as you can see, we've got this whole complex chain of things where, as opposed to just like a simple ion channel. But the end result is very similar in the sense that just as if we had an ion channel that was selective or permeable to sodium, we now have an influx of sodium entering our cell as a result of this whole cascade. So comparing this to our umami, it's actually very similar. One of the only things that we're going to see being different is actually with the receptor itself. So we're going through and we're looking at it all, and really the only things to keep in mind here that are different are, again, the fact that this receptor is different. You can also see over here that we have a tail that's attached to our receptor. And again, this is just like emphasizing the fact that these are two very different receptors, and even though their mechanisms are so strikingly similar, especially when you compare them, they are not the same receptor. And remember that also that the tastes that we're perceiving are very different. Here with the umami, we're getting a more savory taste, right? And it's also very biased, or not biased, but it is really receptive towards proteins and amino acids, right? So that's a lot of what this is. Versus up here with our sweet, we have sweeteners and sugars. That is what we're going to sense when we have this activated. Sweeteners and then sugars. So as similar as these two are in terms of the actual mechanism, it's interesting because we actually taste very different things. So it's something to keep in mind that even though the mechanism can be similar, we're might end up tasting very, very different things.